Hello guys, welcome back to a fresh new topic, that is history of plant biotechnology. First one is 1902 Land. He is the known as father of plant tissue culture. What do you mean by plant tissue culture? Plant tissue culture is a in vitro culture of a plant cell, tissue or whole plant under a controlled nutritional and environmental condition obtained to produce a clone of the plant. Robbins, 1922, he experiment on in vitro culture of root tips. Liebig, in 1925, use of embryo culture technique in interspecific cross of linseed. What do you mean by embryo culture? Embryo culture is a component of in vitro fertilization where in result embryos are allowed to grow for some time in an artificial medium. What do you mean by interspecific cross? Interspecific cross means same genus but different in species. White. Next is White. In 1934, he successfully cultured of tomato root. Next is Van Overbrick. In 1941, use of coconut milk, he first time cultured the datura embryos. In coconut milk there is a component is there who is specifically used for the cell division factor. In 1952, Morrell and Martin use of various free dahlias. First application of micrografting. What do you mean by micrografting? In horticulture crops, in vitro technique is a grafting. It is the grafting of rootstock, establishment of rootstock and shoots. Moore et al. In 1954, first plant regenerated from a single cell. It is the first biotechnological application in plant cell, single plant cell. In 1956, K. Korenberg et al. He synthesis of DNA in a in vitro fertilization. What do you mean by in vitro? In Latin, in vitro means inside and vitro means glass. The inside the glass is known as in vitro. Next is Shook and Miller. In 1956, he discovered the regulation of organ formation by changing the auxin and cytokinin ratio. Retain and Setward. In 1956, in 1959, regeneration of embryos from callus, clumps, and cell suspension of carrot, Dacus carota. It is an weed. What do you mean by callus? Callus is a unorganized growing mass of parenchymatic cells. In biochemistry, the formation is induced for the plant tissue sample after the surface sterilization and planting into a tissue culture medium is known as callus. In 1960, describe the enzymatic degradation of cell wall. In 1962, Murex and Sug development of Murexine and Sug nutrient medium. It is in nutrient media which is developed by the Murens and Sug 1962. In 1946, Guha and Maheshwari production of first haploid plant from the pollen grain of Datura. In 1968, Hedzi Koran awarded Nobel Prize for the genetic code of Hedzi Koran ETL in tRNA. In 1970, Smith discovered the first restriction of endonuclease of hemi hemophilios from homophilios influenza. It was later purified and named as Hind 11. In 1978, Micheller he described the pomato. He hybrid between tomato and potato and resulting pomato. In 1972, Carlson ETL first report of interspecific hybridization through the protoplast fusion between two specific of nicotina. In 1980, Eel Lilly and co-workers and its co-workers commercially production of human insulin through genetic engineering in bacterial cell. In 2001, Human Genome Project is started for the human genomic sequence. Some terminology used in plant tissue culture which are useful for that completion of biotechnology. What do you mean by organ culture? Organ culture, it is associated to a plant organ like root, tip and leaf and flower parts, etc. What do you mean by cell culture? Culturing of single cell, a small group of similar cells is called cell culture. What do you mean by aseptic culture? Raising of culture from tissue of an organ after elimination of bacteria, fungi and microorganisms. What do you mean by suspension culture? Suspension culture is a cell aggregate in liquid medium. Batch culture, cell suspension grown in a fixed volume of liquid medium. Callus, a tissue arises from unorganized cell either in culture or in nature. Subculture, what do you mean by subculture? Aseptic transfer of a plant parts culture to a fresh medium. Passage time, the time interval between two successive subculture is called passage time. Next is clonal propagation. What do you mean by clonal propagation? It is the propagation of asexual multiplication starting from a single individuals. What do you mean by micropropagation? The production of a mina, true planting materials, in large number of vegetative multiplication through the growth hormone regeneration is called micropropagation. What do you mean by hybrid? A plant cell which having cytoplasmic hybrid produced by fusion of protoplasmic plast. Cytoplast, sorry. What do you mean by protoplast? A single cell with their cell wall stripped of a cell without a cell wall is called protoplast. What do you mean by cytoplast? Cytoplast, it is nucleus included protoplast. Nucleus included protoplast. What do you mean by heterokairiosis? Carrion, heterochiron. What do you mean by heterochiron? A cell with two or more nuclei of similar genetic makeup is known as homokaryon, and different genetic, it is known as heterokaryon. Synkaryon, the hybrid cell produced by a fusion of nuclei, is heterokaryon. Heteroplast, what do you mean by heteroplast? Cell containing foreign organelles is known as heteroplast. Genetic engineering, manipulation of genetic architecture of an organism of DNA level or molecular level is known as genetic engineering. Genetic engineering is the architect of DNA or RNA level with manipulation of genetic materials or molecules is known as genetic engineering. Thank you for watching.